Yeah.
Okay, so welcome back to uh, the series of 4 lectures on quantum biology, and today we'll be looking at um, two topics which are, I would say, central to this issue. And the first one is metabolism, or how energy is being transduced in living systems, and what that means in terms of quantization. And the second will be something related to it, but quite different, namely how the brain works and whether we can find some aspects of quantization in the human brain. There's a lot of mysteries related to it, but we'll try to use physics to unravel some of the issues. So starting with energy. Um, so I'm trying to reduce some of the noise on the screen that I see. In general, we can talk about two sources of energy for living systems, the main ones, and these both of them actually imply quantization. The first one is, of course, light, photosynthesis in plants and some bacteria and some algae, and the second is nutrients, mainly glucose and some nutritional compounds, or that in the end become transformed into glucose, and glucose then is used to produce ATP molecules or GTP molecules, which are the usable forms of energy, usable quanta of energy, if you will. Systems such as us require for all molecular level processes. And so this is the what I refer to as the neglected player, mitochondrion. Light, of course, it's in the form of photons, quanta of electromagnetic energy, and then this was actually the first and the most well-studied phenomenon that can be ascribed to quantum biology. Photosynthesis is a quantum process. Okay. Oh, and I'm going to use the red ball pen, and we'll go in this way. So the question of energy consumption or transaction, in fact, because we transduce energy, we don't consume, we just turn one form of energy into another, has a very long history, and is can be traced back to some very famous scientists, Laplace and Lavoisier. So we are talking about a quarter millennium ago, 250 years ago, roughly. Um, and these two scientists introduced the concept of metabolic rates. So the amount of energy transduced and transformed into heat, as well as useful work by different organisms. And the question then was kind of refined by Rubner and then later on Kleiber, in terms of how these amounts of energy can be related. So energy transduced per unit of time, which is power. In physiology, the term used is metabolic rate. How that is related to the size of an organism, and some other things such as lifespan. So these systematic studies led to some very interesting conclusions, namely that the energy or the metabolic rates, to be quite specific, do not scale linearly with size, but in most cases. So as opposed to, let's say, predictions or some intuitive understandings. So this resulted in the so-called the formulation of allometric scaling laws, such as this. So they actually apply to some properties, not necessarily metabolic rates, maybe even heart rates and respiratory rates, and let's say lifespan, things of this nature. Any sort of scaling of a property Y with the weight of the organism across different species, and within the species across different individual representatives. So so the prefactor of alpha is just specific to a given species, let's say. And then what's interesting is this exponent beta. If it was linear with size, then beta would be 1, and there would be no major question or mystery. However, as you can see below, this is not the case. It's very seldom the case that beta is 1. So there are different measures of metabolic activity. I don't really want to get into the details because this is not the main topic in this lecture. But you know, you have something called the basal metabolic rate and the field metabolic rate for animals, and also the maximal metabolic rate. So so these things, all of which, by the way, scale. They may have different coefficients alpha, but they will scale for a given set of organisms with the same exponent beta. So starting with, because as I mentioned to you, metabolic rate is the amount of energy transduced over time. So it's first of all important to find out what a scaling law applies to time, physiological time, which is cyclical. So let's say time could be the lifespan, or it could be, let's say, heart rate, you know, anything to do with the passage of time. It could be cyclical, or could be, you know, linear. So that's the exponent that is found for physiological time. 1/4 beta is 1/4. And then when you look at the metabolic rates themselves, and especially basal metabolic rate, then you have these kinds of sets of exponents which are characteristic of different organisms. Unit cell have very clear 3/4 exponent, which is kind of dominant in all other all other species. But for plants, there is a greater variation between 2/3 and 1. For animals, between 2/3 and 3/4. So you see everywhere you see there's something either 3/4 or 2/3, and that drew attention of scientists. So we will look at these characteristic exponents of 1/2, 3/4, and 2/3. I think it's not shown here. 2/3, 1 and 1. Okay, so how do we explain this? And there are some specific issues, like there are variations in this proportionality constant alpha. That probably has to do with the specific way of transducing energy. You know, birds versus mammals. You know, what kind of digestive processes are used, and how efficient they are, and what sort of nutrients the animals consume, etc., etc. So that's not my main interest. It definitely is an interest of zoologists and biologists and physiologists. But my interest is in relation to quantization, and there are some interesting observations to begin with. There are different geometrical arrangements of energy transmission producing organelles. So there is the bacterial membranes with different enzymes, and then you have especially in plants, you have of course chloroplasts. This is in this case the energy is that of photons, light, and it's harvested and used, funneled and used for the production of glucose in the process of photosynthesis. In higher animals, we have the complex situation with mitochondria. Mitochondria, shown here, are very complicated organelles, about 500 nanometers long or so, and they have the inner and outer membrane, outer inner membrane. The inner membrane is where most of the action is, and the action involves, as shown in this little cartoon here, involves 5 membrane protein complexes through which electric charges are transferred, protons, in both directions, but mainly toward the intermembrane space. And there's the in addition to that, importantly, you have the electron transfer chain. So it's perpendicular to the movement of protons or positively charged units. We have negative transfer of electrons, which is driving the process, and it's critical. This part of the process has been demonstrated to be quantum tunneling. So these electrons tunnel across the complexes, and as they do so, this is coupled to proton transfer. And in the end, basically there is ATP synthesis. ATP molecules are quanta of biological energy, and they are produced and released outside of the mitochondria by these ATPase enzymes, which are also very complex. This whole machinery is just amazing how complex and intricate it is, and how efficient as well it is in its functioning. So from ADP, the low energy version, adenine diphosphate to adenine triphosphate, high energy ATP, low energy ADP. A phosphate group is attached, and that carries this extra amount of energy. It depends on the concentrations and so on, roughly something like 60 kilocalories per mole. It's this amount of energy is large compared to thermal noise, thermal energy, KT, which is, for comparison, this is free energy because, let's say we assume it, we and mammals and many other living organisms function at the physiological temperature, but even if it's at the ambient temperature, KT is on the order of 0.6 kilocalories per mole. So that's the basement level of the biological energy scale. Anything which is below this can be ignored as noise, and anything which is at least, I would say, 2 KT is meaningful. In the case of ATP, ATP is about, again, depends on the concentrations and so on, but roughly speaking, 60 kilocalories per mole, which is about 100 times. And why is it significant? Because that tells you that processes which involve ATP as the energy unit are deliberate, and the amount of thermal energy is negligible, about 1%, compared to the amount of energy in ATP, meaning that that the biological system cannot be affected by thermal noise. All right, so and then so this is the first two aspects of metabolism which have to do with quantization. Number one is units of ATP, and in some cases GTP, which is guanosine triphosphate. It's an equivalent, has a bit less energy than ATP, but let's say we focus on ATP as the main biological energy quantum, and this is integer. So units of, like in early quantum theory, remember when I said in the first lecture, electromagnetic waves are quantized in units of h omega, Planck constant times frequency, 1, 2, 3, 4, 5, but not 1.7 or 1.6. The same here with ATP, we can have integer number of ATP molecules, and many processes such as transport inside the cell, motor transport, and actin-myosin complex and contractions, which lead to the muscle contractions and the work of our muscles. They all use ATP for change of state, and that's so that's number one. Number two, I mentioned to you this electron transfer chain where there is real quantum process of tunneling of electrons from state to state on these protein complexes. The actual geometries of the various metabolic energy factors, if you will, depending on the system, unicells or chloroplasts or mitochondria, they vary, and that's kind of significant, as you will see, and also across the body. You know, from cell to cell, you build a certain geometry. And so the term quantum metabolism was introduced or coined by Lloyd Demetrius, actually my colleague and sometime collaborator. This kind of theory integrates other phenomena, namely, the first one is chemiosmotic coupling between the electron transfer process and ADP phosphorylation or ATP production. This is Mitchell's explanation of how mitochondria work. That was a Nobel Prize in medicine. I don't remember the year, but late 1960s, I believe. Then the second is the storage of metabolic energy in vibrational modes of various molecular components of the membrane. And the third assumption or building block of this quantum metabolism by Demetrius is quantization of the energy in the form of synchronized oscillations. So this has been demonstrated by experiments on cells that the metabolic enzymes across the organism are synchronized, and that leads to efficiencies. So I showed you this before in a small version. This is a bit bigger, and you can see here a number of reactions in this electron transfer chain, part of the oxidative phosphorylation phenomenon, which is the highly efficient but very long process of transferring molecules through various parts of the mitochondria. And here in the blue is shown the electron transfer chain pathway, and the red arrows indicate the proton transfer across these complexes. And finally, again, you see here that's a rotating motor protein, FATPase. Okay, so that's low high energy electrons travel from ADP and a phosphate to ATP, and ATP is used to perform metabolic rate. Okay, so what the theory 1970, so Mitchell 1970, so I was a bit off. The second, that's three, let's say Nobel Prize, either Nobel Prize winning or Nobel Prize worthy building blocks for this quantum mechanical theory of metabolism. I mentioned the chemiosmotic coupling of Mitchell, where ADP phosphorylation, ATP production is coupled to the electron transport, which is a quantum tunneling. The second is I think I mentioned last time briefly, of electromagnetic oscillation cause synchronization across an organism, at least a cell. And the last one is strong analogy, or the use of Debye's theory. I also mentioned to you maybe in the second lecture, Debye theory of quantization of phonons, not photons, phonons in solid state. And that was due to a quantization of waves. So phonons are mechanical waves that propagate across a crystal, for example. Here, Lloyd Demetrius used this mathematical model that Debye proposed, and actually improved on Einstein, to represent coupled oscillations of Venza enzymes in metabolic reactions, just like coupled oscillations of molecules form phonons. So these three building blocks put together form the foundation of quantum metabolism, and yeah, and these oscillations can be quantized. And that was, I'm not going to go through the math too much, a little bit, but this is the assumption that the nth energy level of metabolic in the metabolic energy production excitations has n quanta, and omega is characteristic frequency for these enzymatic oscillators. They go in cycles. This is the original paper that Lloyd Demetrius published, where this model was proposed, and not only proposed but also used to explain what I started from, namely these allometric scaling laws. Okay, so again, this is a bit mathematical, so I'm just going to skip through it, but some of you might be interested. This is how you calculate this is the Bose-Einstein model of average energy calculations for quantum oscillations. And for the metabolic network, you use this like so, and overall the oscillators, and you go into the space of waves, and there's a little bit of algebra which you don't need to pay too much attention to, but it's a very rigorous calculation. And so you can integrate it. So that's the energy over all the frequencies which are allowed within the model. This is the number of these frequency density of states. It's called the number of such modes, and this is the averaging process. And you obtain for the energy, you obtain this kind of relationship with the characteristic cycle time. And what's very interesting here is that there are two regimes: the quantum limit, when the cycle time is below a certain characteristic value, and that leads to this formulation. D is the dimensionality of space, so it could be 1, 2, or 3. Why would that be? Well, if you have oscillators, let's say linearly positioned in some very quasi-linear arrangement, or you can have a plane distribution, planar, maybe in epithelial cells, or in three-dimensional organization of metabolic enzymes in some, solid organ, then you might have different scaling laws, and that enters into this dependence. Now, the metabolic rate is basically energy is proportional to U divided by the U dt, d tau. So you have to do calculate the derivative, and you obtain these corresponding scaling laws. P is the metabolic rate, and W is the weight of the. Remember, there was tau scaled with weight as power 1/4. So anyway, when you do this, you obtain the final result, namely the metabolic rate. If the time characteristic cycle time of this process is much greater than this critical value, then it's linear. But otherwise, you have weight to the power d over d plus 1. Interesting, right? Because d equals 1. And d equals 1, the exponent is 1 divided by 1 plus 1, which is 1/2. When d equals 2, then you have 2 divided by 2 plus 1, which is 2/3. That's the exponent which we called beta before. So 2/3. But the dominant case is when dimensionality of the system of oscillators is three-dimensional system. Then the exponent is 3 divided by 3 plus 1, which is 3 divided by 4, 3/4. And that's the characteristic exponent which is seen in all the cases that I've mentioned in the beginning. And that's the Kleiber scaling law for allometric processes. Okay, that's again written down here. There's also some, because enzymatic reactions can be activated and at different temperatures have different rates. So there's that from Mitchell theory. But the main result is this: the beta exponent is related to the dimensionality of these coupled oscillators, metabolic oscillators, and that's it's completely consistent with the observations. I will just very briefly, because there's a lot of information here on this single slide, say that with this you can define something just like in the very first example of quantization. Remember, Planck quantization of electromagnetic radiation was E equals Planck constant H times F. You can also define a similar relation for metabolic oscillators. So electromagnetic waves are oscillators of electric and magnetic fields in space and time. Here we have the frequency F is the frequency related to these oscillations of metabolic enzymes. And the question is, what is kappa? This would be what we call the biological Planck constant. H was physical Planck constant. Now this could be the biological Planck constant, and you can go through this reasoning here very briefly. If you convert something like the energy consumption of an average human being, which is about, let's say 2,000 nutritional calories, which are actually thousands of physical calories. So 2,000 calories a day, let's say 1,500 to 2,000, depending on the sex and the body size and so on, roughly speaking, 2,000, you obtain this number of ATP molecules per second, which is astounding, right? 10 to the 21st ATP molecules per second. It turns out to be the turnover of ATP molecules in the human body is equal to the body weight every day over 1 day. So that's our normal energy requirement. Now, this is the number of cells in the human body. Each cell has roughly 1,000 mitochondria. So overall, we have something like 3.5 times 10 to the 16th mitochondria. And then this is the amount of ATP, so biological energy quanta produced per mitochondrion per second, 30,000 or so. And okay, this is the conversion of 1 molecule of glucose into 38. So you can convert this into how many glucose molecules each mitochondrion requires for this. Each glucose molecule produces 36 to 38 molecules of ATP. And then there are these ATP synthase enzymes, which are these oscillating enzymes I mentioned to you before, and they operate about 600 ATP molecules per second, and there's about, therefore, 50 ATP synthase enzymes per mitochondrion. And the rate of oxidative phosphorylation reaction per enzyme is about 1,000 cycles. So this is the frequency, characteristic frequency is 1 kilohertz, 1,000 cycles per second, or a millisecond is the characteristic time, turnover time. So if you do all this calculation, you obtain something like, for this kappa, biological Planck constant, is 10 to the minus 24th joules. This is the physical Planck constant. So they're off by about 11 orders of magnitude. This is the ratio, 1.8, 10 to the 11th. Interestingly, this is roughly, it corresponds to the number of atoms in a cell per mitochondrial, I call it sphere of influence or region of interest. So roughly speaking, that corresponds to the ratio to the Planck constant. Planck constant, remember, originally was related to, let's say, a single atom transition between electrons in a single atom. Here we have oscillations or transitions between states in a mitochondrion, and that is how many atoms this and this part of the cell contains. So it makes sense. It scales very closely with the size of the object, and therefore also scales with the frequency, which is much slower than atomic level frequencies. So that's kind of a interesting and illuminating, in my opinion, calculation. Now, so that pertains typically to mitochondria, which is animal cells, mammals, us. But there's very strong and well researched topic of quantum coherence in photosynthesis and photosynthetic systems. And this goes back to not quite 20 years ago, but coming on soon on 20 years, which was studied by these groups, both experimentally and theoretically. And it was determined that in photosynthetic complexes that are so-called FMO complexes, I'll show you in a second what they look like, light creates excited states of electrons in these molecular complexes, and these electronic excited states are coherent or correlated across the entire complex, which is several nanometers in size. So photosynthesis, kind of schematically captured here, is light energy from the sun striking the surface of the leaf. There's a chlorophyll that captures it, and the energy transformation and magic happens, and then we obtain this energy to with carbon dioxide being needed, being necessary for this reaction, plus water, plus energy, so giving you a molecule of glucose. Okay, so sugar. Okay, so that's the and that's the structure of the complex, which captures this electromagnetic energy of sunlight. So FMO is the name of the complex, Fenner, Matthews, Olson, after the names of the people who discovered it, and also is present in bacteria, like those bacteria and bacterial chlorophylls in the hot springs of New Zealand. It's a thoroughly complicated structure, and the process is shown here in schematic representation. And again, and that's the structure of the active complex, which is involved in the capture of light quanta and creation of these excited states of electrons called excitons. There's 7 identical molecules, these heterocycles, and they are all quantum correlated. If you excite one of them, this energy is quantum mechanically, it's a wave function that is spread over this entire complex, and it's it could be more than one at the same time, so we can have several quanta activating these complexes, and then the energy is being transformed into excited states of electrons, which then produce or are being funneled and utilized by the plant to create this glucose molecule. So that's another representation, and these papers explain that the quantum mechanical mechanism involves quantum search algorithms for reaction centers and capture and distribution of the energy. So these systems also exist in algae, and cryptophytes are the algae that use it. And by very careful experiments in the laboratory, it was also shown that the wave functions are entangled in this complex, and this was done by quantum beats being observed by using laser light and measuring the time scales involved, which are not very not very long, hundreds of picoseconds or so, but this is enough to perform useful work and transfer the energy to the most effective sink. Excuse me. Now, so let's switch gears a little bit. I have the last half of the lecture to show you something similar, but in a completely different system. And I'm going to talk about microtubules for the reason that microtubules have been touted as another subcellular system that is capable of creating quantum excitations, just like this FMO system in chlorophyll, in plants and in bacteria and algae. But this is now we are moving to, well, microtubules are in plants as well, by the way, but we'll be focusing on mammalian cells, especially neurons, in fact, human brain. We'll get to it. So here's a microtubule. That's a cartoon of a cell with a nucleus, and tubulin is the building block of microtubules. This is a mitotic cell. Cell is dividing. You have the chromosomes in the middle plate, and microtubules connected to connecting the centrosome and the kinetochores, orchestrating cell division with very beautiful perfection in normal situations. That might also beg the question whether this orchestration has something to do with quantum. And I don't think that has been explained by anybody this way, but I think it's an attractive hypothesis to test. So microtubule cytoskeleton plays major roles in various cells, but we will now focus on non-dividing cells, neurons, in fact. And here you see the same microtubules as these black line segments connected by thin lines, which are microtubule associated proteins called MAPs. And as you can see, microtubules, we call them MTs, microtubules fill both the axons of neurons, the soma, and the dendrites, and they perform such functions as, of course, structural stability, growth cones. And so when in early development neurons grow, the microtubules guide the growth. And then, of course, transport inside the neurons is on microtubules through kinesin and dynein motors. Each motor consumes one molecule of ATP per one step. So the process of transport is completely quantized. That's another thing that should be stressed. And microtubules themselves, when they grow, they consume one molecule of GTP, with analog of ATP, per one addition of one unit. So as you can see here, quantization is at the level of structure formation as well as the level of transport. Now, about 30 years ago, so Roger Penrose, who got a Nobel Prize in Physics in 2020, and then with later on, well, he wrote these famous books on the enigma of consciousness, The Emperor's New Mind, and forget the title of the second book, but anyway, opened the discussion that consciousness, and in particular, and sort of human mind operates on quantum principles. He gave very strong arguments that classical explanations don't cut it. They cannot really explain how the human brain works. And I encourage you to read these books by Roger Penrose, because they are incredibly powerful and interesting statements about the state of our knowledge in this area. And then later on with Stuart Hameroff, an anesthesiologist from the University of Arizona, suggested that actually the processing of information in the human brain involves quantum computations in microtubules. So that's why I'm talking about microtubules. When you look at microtubules, they're formed from these proteins called tubulin beta alpha heterodimer. So it has two units, but stably bound together. And there's a particular amino acid residue in tubulin, which is called tryptophan. And tryptophan is unique out of the 20 amino acids because it can be excited by ultraviolet light, near visible, but not quite. And there's 8 tryptophan residues in this building block, 4 and 4 in beta pathways shown here and here on the surface of microtubules, which can be used just like in the photosynthetic systems for tunneling of excitons or quanta of electronic excitations, if there is a source of electromagnetic waves that can excite them. Okay, so here is just a representation of this chromophore network in tubulin of tryptophans. And remember, there were seven molecular complexes in the FMO case for chlorophyll. And here we have 8, so it's somewhat a similar arrangement. And here are distances between them in angstroms. And some of them are a bit closer, quite a bit closer. There are some rules in quantum mechanics that the tunneling distance and exponential decays over the separation between two centers between which they can travel, and these numbers are sort of within the range of possibilities, especially here, the shorter ones. There are some longer ones. But at any rate, you can see them here. What's shown here is this protein, tubulin heterodimer, which is the building block of microtubules. And now we can imagine interactions between them. I'm not going to get into this nitty-gritty of quantum mechanics, because that's not the right course in maybe a graduate level of biophysics or quantum theory that would be correct. But here, this is a Hamiltonian energy representation of the dipole-dipole interactions. These tryptophans have dipoles, and the dipoles interact. This is the interaction matrix. The important thing is that they can be excited by light in the UV range between these numbers, 260 and 305 nanometers. And once you excite them, they can interact and do something like I showed you for chlorophyll. And these excitations, as you can see here, were predicted to last picoseconds, maybe not hundreds, but picoseconds. Short, but maybe there's some way of making them last longer. And in fact, this is a computational prediction, which we published together with my student, Travis Craddock, who is now a professor at the Canada Research Chair, in fact, at the University of Waterloo. So I'm very proud of him. He's done very well for himself. But recently, experiments were performed recently, which is in the last 2-3 years, and I was involved in these experiments that were funded by the Templeton Foundation, and we had Greg Scholes from Princeton, Aristide Dogariou from the University of Central Florida, and this is the example of delayed luminescence that was studied by the group of Professor Dogariou. Namely, you shine or illuminate microtubules in a sample in solution over a period of 2 minutes, and then you observe the re-emitted light or photons from the system and see how long it lasts. That's the sort of technical representation of the laser system used. There was excitation. This is the cross-sectional area, 3 by 3 millimeters squared. The intensity of laser light, which was not very high, that corresponds to electric fields of about 200 volts per meter, rather weak. And then there's a detection apparatus, basically photon counting, and you observe it for tubulin solution, microtubules in solution, microtubules with tubulin. We also did microtubules with anesthetic molecules for the reason that I'll explain a bit later. And you basically observe the intensity of re-emitted light. Now, this is optimistic, but it turned out to be actually milliseconds. It's called delayed luminescence. So this protein in solution and microtubules are basically assemblies of this protein tubulin. So in the microtubule, you have 13 rings around the cylinder and hundreds of layers. So if it's a microtubule that has 10 micrometers length, then this structure will contain something like 16,000 units of tubulin or so. And they create when you eliminate with this light, they then re-emit it, but very slowly, so that there's something happening inside, trapping of this light and maybe transfer. Remember, for chlorophyll, photosynthesis is actually the fundamental process of energy capture, energy of sunlight capturing, being captured in the food chain on this planet, and really the most fundamental process without which life on Earth would not evolve and continue. So that's super important, in my opinion. And here you have something similar potentially happening inside microtubules, and microtubules, as you saw a few slides ago, form densely packed structures in neurons, and neurons are used for our understanding and processing of all the information about the world around us. So that's, in my opinion, very powerful as a proposition. Interestingly, one possibility of where these photons may come from, of course, we are not illuminating to the same degree as plant leaves are illuminated by direct sunlight, but neuronal microtubules are inside the cells so that they cannot obtain this sort of level of illumination. But mitochondria, which are we talked about mitochondria a lot. They are the energy producing organelles, ATP molecule producing, but at the same time, one of the byproducts of this process is emission of photons. And lo and behold, mitochondria in non-dividing cells, or in non-dividing phases of the cell cycle, interface associated with microtubules. So quite plausibly, the emission of mitochondria, for as long as they function, could feed microtubules with the electromagnetic energy needed for all kinds of electronic processes, a deeper layer of information processing. Okay, so this leads us to the sort of last part of my lecture series. We now zoom out a little bit and address the questions regarding our brain, what makes the brain special, and what is consciousness, and where is memory stored. All of these are open questions, and there are debates about how to define this, or is memory, and what these things we can answer. What is the computational power of the brain? And that's very intriguing. And then how much information can be processed in the brain? Is it comparable to a computer or not? And things of this nature. And then you will see that the power consumption of the brain with the potential information processing numbers indicate incredible efficiency of information processing compared to that of the modern day, the latest types of computers we use from our latest technological advances. I want to get you interested in these conferences if you're interested in consciousness.
So
Stuart Hammer.
there's a question from Hish.
Yeah.
Hish, did you have a question?
Yeah, so it's very interesting, and I like the way you're basically like bringing up this topic, and like it's basically everything that you've been talking about is leading up to this. And in a way, I've been thinking about a lot about consciousness as well. So I have some like questions, if that's fine with you.
Sure.
Should we do it now or wait for a few minutes? Because I'm not going to be needing much more time to conclude this.
I see. I can wait till the end.
Yeah. Good. Okay.
Okay. Then I'll be able to complete what I prepared, and then we can talk. So I just wanted to say that these conferences have been going on for about 3 decades now, and they alternate between Tucson, Arizona, and some other destination. In 2023 was in Sicily, Italy. Actually, this year, and not this next year, in 2025, it'll be in the first week of July in Barcelona. So nice, interesting location. Maybe not now with the flooding and all that, but by July, I think it should be dry. Barcelona. Okay, so you can look it up on the website to what they call the Science of Consciousness, a gathering of people that are probing these deep issues. So some skeptics, I guess, say that the brain is not processing any information. And this is by Robert Epstein. You can post it, and you can find the post here. Brain is not a computer. I completely and utterly disagree with this. Of course, brain is not a computer, but it does information processing like computers and maybe better. And this is kind of naive. I wouldn't call it stupidity, but naivete to try and find a copy of Beethoven's Fifth Symphony in the brain. I mean, it's not a storage of symphonies. It's a processing and information storage and retrieval, but not like that. So let me just say that the human brain has amazing numbers characterizing it. So roughly 100 billion neurons in an adult brain with about 10,000 synapses per neuron, which leads to about 10 to the 15th synapses. And if even at the sort of modest rate of 10 impulses per second, so every 100 milliseconds, an impulse or activation event takes place, then you can reach something like 10 to the 16th synapse operations per brain per second, which, by the way, would outperform the blue gene, which has 100,000 or more CPUs, the supercomputer built by IBM. So that's already kind of impressive that our brain may potentially operate at a higher information processing rate than the supercomputer. So back to this. But even more amazingly, the energy consumption of the brain is minuscule compared to that of the supercomputer. 25 watts, about 25% of the human energy requirement for the entire body is used by the brain. So a large demand, and if demand is not met, there are some issues. We know that glial cells provide neurons with the required energy balance, but here's what blue gene requires: 1.5 megawatts. So that's what 100,000 roughly times greater, and that's kind of mysterious. And this kind of raises questions. Maybe is this actually, as Roger Penrose and Stuart Hammer would probably agree with, therefore, even on the issue of energy consumption, a quantum computer is more powerful than classical and has lower energy consumption. So maybe at least part of it is working at a quantum level. And then also consistent with what Penrose and Hameroff have been talking about, collapse of the, if this is quantum, a collapse of the wave function in the quantum mechanical parlance is potentially a realized thought, or a moment when we either verbalize or realize a thought that until that moment is in a superposition of possibilities. So that's kind of a direct either a realization or metaphor between quantum mechanics and neuroscience. Okay, and are there any quantum mechanical limits on the physical predictability of the brain? These are the questions. And then, of course, the key one is the role of temperature, which in normal quantum mechanical systems would be the killer of coherence and quantum states, but somehow maybe Mother Nature has found a way of overcoming this thermal distraction of quantum coherence. And then this also leads to some implications for artificial intelligence. As you probably know, for now, I don't know of any artificial intelligence algorithms that utilize quantum processes that would actually empower it incredibly. Okay, so oh yeah, that's the book that I referred to, Roger Penrose's The Emperor's New Mind, and this is now 30 years old, I think, something on this, maybe 25 or at least, and I was prescient and opened a lot of avenues for investigations and discussions. So in this Hameroff-Penrose model, which is called ORC-OR, or orchestrated, I'll explain why this objective reduction. So orchestrated means that all these tubular units seen as qubits in the language of quantum computing. So this is a superposition of two states, but they collapse. The wave function of this entire microtubule or even neuron collapses as a result of orchestration or internal interactions. And this is all objective. That means it's not it's well, I don't know if I can explain very well the word objective, but it really has to do with some other assumption, yeah, which is a little bit technical. It means that the superposition state actually is between is interacting with itself gravitationally. This is a difficult assumption to explain. I'm not going to spend too much time on it. It simply means that the system itself decides when and how to collapse. It's not triggered, it's not moved by external circumstances, so to speak, but stochastically or unpredictably decides to collapse into a particular solution, which is a bit like I would say a door, an avenue toward free will. So that's why it's objective. And so here's kind of a scheme of things. The tubule and dimer schematically, a microtubule being formed. This is each of these protofilaments. And then you have the connections between microtubules inside the neuron. And by the way, there is circumstantial evidence that a lot of things happen in the wrong way if some of this goes bad. For example, these connections maps, if they detach, then that leads to neurodegeneration, and almost every neurodegenerative disease is related to the malfunctioning of the microtubules or the microtubule interactions with motor proteins. And you have Alzheimer's and Parkinson's and Lou Gehrig's traumatic brain injury. All of it is related to microtubule pathologies, let's put it this way. Not necessarily the only thing happening in these in these terrible neurodegenerative diseases, but one of the key things. Okay, so this is, I'm going to skip because it's kind of standard neuroscience. Signal propagation between neurons and synaptic connections and neurotransmitter release and activation and the postsynaptic connection and action potential traveling along axons. But there is some microtubules that are deeper inside axons and inside dendrites. So what current modern neuroscience is focusing on is the outer layer, and we are looking inside, and that's why Penrose and Hameroff believe that the quantum, and maybe it's shielded by the membrane and free from disturbances from the outside, therefore could be stable quantum mechanically. So this is, again, inside the dendrites, you have specific arrangements of microtubules, which are actually anti-parallel. Microtubules have the plus end and the minus end, and in dendrites of neurons, uniquely, they are ordered in an anti-parallel organizational mode. That means every other microtubule is flipped in the opposite direction to the previous one, which might lead to something like back propagation of signals, or maybe some backward and forward propagation of signals and feedback loops and all this. Okay, so yeah, this is what I mentioned. All these diseases, stroke, brain injury, neurodevelopmental disorders, neuropsychiatric disorders are related to issues with microtubules. And if we buy into Hameroff's and Penrose's theory that microtubules are the quintessential information processing units, computer chips, if you will, performing some computations like this, Hameroff and others showed signal propagation in the form of cellular automata. There may be some logical gates, etc. Then this would all make sense. That defects in these structures or some kind of destructive process that deteriorates the connections may change the possibility for memory storage and information processing. So first of all, and I'll be done maybe in about 5 minutes, but I think this is important. First of all, if you just count something that we know, how many possible states each of these, then you do the math and extrapolate to microtubules and how many microtubules a neuron will have, a few thousand, and how many neurons we have, 100 billion, you start getting some astronomical numbers of possibilities. And these are the states that we know exist, and they can contain information. So C-termini states, C-termini are these charged protrusions of each of the monomers in a tubulin dimer, and they can be at least in 2 states, stretched out or bound to the surface. It's like flipping a switch from bent to straight, and you can encode 0 or 1 in it. There could be also electron hopping. We know that there are conformational changes triggered by GTP hydrolysis, again, energy states, and some other ones. But even if you just count these ones, you end up with something like a gigabyte of information per neuron and 10 to the 20th bits of information per brain that can be stored in microtubules this way. Now, how fast these states can make transitions depends on which ones, of course. But of course, that's optimistic nanosecond transitions, which conformational states of proteins support. That would be a phenomenally large number of operations per second, 10 to the 28th. That's I would say that's impossible, but upper limit for sure. Now I did some more realistic estimates of this number of operations based on somewhat more realistic assumptions, and you still get the number of operations per second could be, you know, something like this, or even higher. Okay, I'll skip this because this is a bit more technical. There's something called the Landauer theorem in information science, saying that it costs energy to erase one bit of information, and namely it's a KT logarithm of 2. And when you take this into account, there's some restrictions on these numbers, but you can still obtain this number, 10 to the 20th of bits per second per brain, 10 to the 20th. Okay, so that's very, very impressive. Okay, so I'll finish now in 4 minutes by postulating where memory may be. I already kind of hinted a little bit, but where memory can be stored, and there's a lot of differences of opinion, but the currently accepted neuroscientific approach is that long-term memory is stored or formed by long-term potentiation, namely the synaptic connections, which I used a lot, are easy to transfer signals, and therefore this is how memory is encoded by easy transfer of signals. Okay, and various people try to estimate this. What is the amount of memory the human brain can store? John von Neumann, one of the people I mentioned earlier, as pioneers of quantum theory, also pioneer of computer science, made probably the best estimate in 1950, 74, almost 5 years ago, 3 times 10 to the 20th, which is almost exactly to the number that I'm going to propose to you. Some other ones, Landauer, that's a different Landauer, Thomas Landauer, psychologist, I believe, very, very, I would say ridiculously small, because today you can have a memory stick you buy for, I don't know, $20 or $10, and we'll have terabytes. Okay, so here is where we, of course, we meaning, you know, Stuart Hameroff, Travis Craddock, and myself, we wrote a paper which proposed that memory is stored in microtubules as a result of something which is known in neuroscience and in cell biology, namely the formation of the calmodulin kinase 2 complex triggered by calcium waves. This complex in the postsynaptic connection forms kind of a transformer-like motor protein molecule, hexagonally shaped, and it is known to phosphorylate microtubules. And we claim that this phosphorylation, that by the way is triggered by some signal from, let's say, visual signal or tactile or olfactory nerve signal, and that's how it works. So you have the at the cell level long-term potentiation, but the molecular level you have phosphorylation of microtubules. We published this now 12 years ago, and we showed through computer simulations that calmodulin kinase 2 phosphorylates preferentially serine molecules on the C-termini of tubulin in microtubules, and can each phosphorylation event, let's say, can be seen as a bit of information, and that's the enzyme. This is the top view of the enzyme, and I showed different graphical representation. This one, C, shows you the charge distribution. So Red is negative charge, blue is positive. And this is the, so this is top view. And here you have the side view. It looks like Stuart Hameroff calls it a poodle or a transformer. It walks on microtubules, and as it walks, it phosphorylates them. So these are the legs. And what's amazing is that it's a beautiful matching, geometrical matching of this enzyme, also looking like a snowflake. And this is a part of the microtubule lattice. And as you can see here, it superimposes almost perfectly to it. And there's also electrostatic matching. So that is based on geometry and physics. And here you can see different representation of the various states on a patch of tubulin on microtubules. And when you count the number of possible states, you can see that there could be 1 gigabyte of information per neuron, but just by phosphorylation of the microtubule surfaces. These are different examples of states you could encode, and you can associate with these some, numbers and binary notation and so on. So this is the paper, brain's memory code, ? And it shows you here this calmodulin kinase enzyme walking on a microtubule and phosphorylating it. And that's the reference to the paper. I will stop now because I would like to hear your question. Thank you.
Thank you for the great classes. Like all the classes, I really enjoyed them. And yeah, so I have like 2 questions right now, but maybe more will come up as we talk. But like the first question is, you mentioned the like collapse of the wave function when basically when we observe something. So basically the observer effect.
Yeah.
I was wondering, what do you think about the idea of panpsychism? The idea that like everything in the universe has some level of consciousness. So like if we are looking at a qubit or like a very small unit of anything, it might have some level of like awareness that like, oh, something is like, so like just like what some ideas of why it might be true or why it might not be true. Just like your opinion.
Yeah, well, I know, yes, you what you said is exactly what Stuart Hameroff, by the way, and Penrose, I think, are promoting as an idea that in fact every molecule, every atom has some sort of proto-consciousness, if you want to call it this way. So quantum superposition of states, and the collapse would be this sort of moment of consciousness. And the interesting part that they claim is that small objects have very short periods of this consciousness event. And the bigger the object, the greater the size of the wave function collectively, the longer this persists. So that would, they claim that would explain that we need so many microtubules in the neuron and the brain, and they come up with numbers like hundreds of milliseconds between conscious events, so very long. In other words, physiologically justifiable. But the rest of the universe, let's say outside of the human brain, smaller things, the grains of sand or whatever, they are too small to maintain some sort of continuity of conscious events. It's all only, very stochastic and very random short bursts of something. Now, what I think about this, I don't know. I'm a physicist and I'm rather grounded, I would say. So for me, I mean, it's already a big mystery to explain how memory works, and I think that there's a lot of convincing even required for the human brain to see this way. Panpsychism, I find it very difficult to accept because there is really no empirical evidence that I do believe that every living system has some sort of proto-consciousness because of the behavior. So if I see behavior which is consistent with information processing, reaction, so even single cells, even bacteria demonstrate some kind of, I would call it, level of intelligence. Okay, so but I don't see any level of intelligence coming from rocks or sand or whatever other objects. So to me, consciousness is an attribute of living systems, and well, so that begs the question: what is life? And yeah, that's my answer.
Awesome. Thank you.
You're
That's
welcome.
a great answer. And you also mentioned fractal resonance and also
Yes,
like randomness. So, like, what do you think is the role of chaos, fractal resonance
yes,
like consciousness? You
yes.
already touched on this with your answer, but just like, yeah.
Yes, so these are, I would say big questions and interesting issues. I think they are more on the level of methodology. I think that number one, fractality or self-similarity or things of this nature is a good idea for explaining how things organize themselves hierarchically. So you may have noticed that I talked mainly about molecular level, atomic level, molecular level, and then neuron, and how does it go up from there? And I think fractality would allow us to see how different levels of organization communicate to the next one. So, for example, how neurons maybe operate at a higher frequency
Hmm.
than brain domains, which are many neurons, let's say millions or maybe more, because the larger the structure, the slower the time scale. So this fractality is to me a very natural way of understanding how hierarchical organization in space leads to hierarchical organization in time. So these time scales, go from atomic level or even subatomic, very, very short, and then slower, and you go to the brain, it's something like, a few hundred milliseconds. This is the time it takes us to formulate a thought or whatever. So
Hmm.
that's for fractality. For chaos also, but that's to me, chaotic dynamics is a signature of the entire brain, because it's a signature of something which is exploring this conformational space of possibilities, and that's probably the best way of searching for or being even ready um, for finding a solution to any immediate problem. If you're stuck in a cycle, it's very difficult to get out of it, right? But if you're in a
Hmm.
chaotic mode of operation, then it's very efficiently organized to find, all regions of the space, searching for solutions, being adaptable and flexible. That's kind of my word salad response.
I see. Thank you very much. I
You
think those are my answers and great answers. Thank you very much.
I'll be happy to answer any email questions as well if you have some.
For sure. Thank you very much.
So do any of the other students have questions? What I've done today for the first time, there's an AI companion that just appeared here. And so it's been producing summaries.
Hmm.
And I think I'm going to send these to Jack, and then if he's okay with it, maybe I'll send selective few to everybody so you can see the the summaries always is trying to summarize the whole period of time, but it does give much more detail on what's happened recently. And for instance, all the early ones mention me, but then at a certain point, it stopped mentioning me. And then it started mentioning Hish, and I don't know, about half the recent ones mention Hish. So it's something I'd like to figure out how valuable are these AI summaries? Should we keep on doing it? Is it just a sort of toy, or is it something substantive? And I think, the students can also help me figure that out. So, yeah, anyway, do any of the rest of you have questions? Um, so I wanted to tell you on Thursday, I'm speaking about transplantation and so the future and AI. And you'll be the first to hear this. It's similar to what I'm presenting at the Mayo Clinic, but it's kind of a practice for that and for the GlomCon session I'm giving on the 17th and so on. So that's what's coming this Thursday, 2 days from now. Anyway, Jack, thank you very, very much.
My pleasure.
Yeah. So, Sarjana, yeah, okay. So
Thank you all. Thank you very
Thank
much.
you
And
also.
again, I encourage you to send me email questions if you have any. I do hope
Okay,
that you do.
excellent. Thank you. Okay, bye-bye now.
Bye.